previous lectures we have covered 5 topics, and these include microscopy or radioisotope technique, chromatographic techniques, electrophoresis and centrifugation techniques. From this lecture we are going to start a new topic, and that is spectroscopic techniques. Now, spectroscopy can be defined as the study of interaction of electromagnetic radiation with matter and its analysis. So, in spectroscopy, the electromagnetic radiation, say light, interacts with the matter in different ways, as we will discuss, and this particular property of matter to interact with the light and the information derived is used for the analysis and characterization of small and large molecules. The interaction of electromagnetic radiation with matter, or as we say spectroscopy, is a quantum phenomenon, or we can say observation of quantum mechanics, and it depends on both the properties of the radiation and the structural composition of materials which are involved in here in interacting with the electromagnetic radiation. Now, different types of spectra obtained or the information which is derived due to the interaction of the radiation with the matter will lead to the understanding of these interactions and development of spectroscopic techniques, which will help in solving many biological problems where one can characterize these molecules and know about them, and likewise solve many biological problems. Little bit into electromagnetic radiation. In very simple terms, electromagnetic radiation can be defined as a form of energy whose origin is the emission and absorption from charged particle. It is like coming out of the matter. So, in terms of classical physics, if we say electromagnetic radiation is mainly produced when charged particles are accelerated by forces acting on them. So, it comprises of both the electrical as well as magnetic components, which are oscillating in directions perpendicular to each other at a fixed intensity ratio, and mutually they are at right angles to the direction of propagation. The energy of EM wave or electromagnetic wave is given by Planck's constant into the velocity of the light divided by the wavelength, that is, or you can say h nu. And let me show you on the screen the typical electromagnetic radiation in a wave form. So, like I said, it has two components. Electromagnetic wave has two components, electrical and magnetic component. So, if I can draw a very specific so there will be two components. One is electric, you can say electric vector, and another is magnetic vector. And if you consider a wave here, so this is magnetic vector at 90 degrees, electric vector, and this is the propagation of the light actually, that is direction of propagation. So, as we depict the wave nature of the light here, what we will have is, if I just draw, supposing this is your one vector or one direction, and if I say this direction as say electric vector here, then there will be another direction which will be perpendicular to this and which can be made in a different manner, likewise actually, and this one here, this is can say, you can give different term H or whatever, and these are the two components of this wave, electric and magnetic components. So, these are perpendicular to each other and then to the direction of propagation here. So, that is a typical electromagnetic wave which has one component which is perpendicular to the two components which are perpendicular to each other. One is electric component or electric vector, another is magnetic component, and they are perpendicular to the direction of propagation. So, that is a typical electromagnetic wave actually. Now, as we said, the energy of this wave in terms of like energy of this wave could be expressed in terms of in this equation here, where you have h is Planck's constant, c is the velocity of light, and lambda is the wavelength of the radiation. Here it has particular this particular could be also like it could be written in this way, that is h nu, where nu is the frequency and frequency is expressed in terms of hertz actually, that is 1 oscillation per second. All right. So, this is typical representation of a electromagnetic wave here. Let us come back. So, what you have is you have Planck's constant, like we have shown you, h c upon lambda. C is the velocity of light in this equation which I have shown you, and nu is the frequency of the radiation. The frequency of the radiation is in reciprocal seconds, and it is expressed in terms of hertz, which is 1 oscillation per second is called 1 hertz. Now, electromagnetic wave, or you can say light, can interact with matter, where it can be either absorbed or emitted or scattered. So, a molecule or atoms, they will either absorb an electromagnetic radiation, or they might emit as they go into a higher state, and when they come back to the ground state, they can emit a particular radiation, or this could be that electromagnetic radiations can be scattered also by the matter. So, the atoms and molecules mostly exist only in discrete energy labels. What does that mean? It means that electrons in atoms or molecules may be distributed, or they reside in ground state, or we say lower level of energy, and they can be excited to a higher level, or we call it excited state, where if exact amount of energy which is equivalent to the difference in energy levels or excited electrons in higher state. So, if it is absorption, then absorption will occur only if there is an equivalent amount, that is the difference between the two energy states is absorbed, and then electron can go to higher or excited state. Likewise, when an excited electron might come back to a lower level, where again it will whichever level it comes, it will emit the equivalent amount of energy, that is difference in energy between the labels. So, light could be absorbed or as well as emitted by a molecule by undergoing transition, and this particular phase where a particular phenomenon where the electron undergoes transition from one state to another is known as what you call transition here, and this transition is between discrete energy labels, and these energy labels and other characteristics are predicted by quantum mechanics. Let me show you on your screen these what does these energy labels mean here. All right. So, when we say energy labels, Like I said, these atoms have a discrete energy labels and they reside in these discrete energy labels, and there is could be a ground state and higher excited states. So, when we say these states, let us little bit understand them. Uh, if I if we can just uh, understand this particular one. All right. Now, if we see these two here, and this is ground state and this is the first excited state. Now, here in this one, so this is electronic state you can say, and this going from this particular one is electronic energy label, another one is in excited state, that is the first excited state of that electron label. Now, within these electronic energy labels there are subdivisions which are known as vibrational labels actually. So, these are called vibrational labels, and these vibrational labels will exist in the excited states also. Here we have shown you the first excited state and they will be in here also. Now, when absorption or emission whatever occurs, say for example, absorption occurs say from the first or from very ground label to higher, if I say it is going up to here, then absorption has occurred, the exact energy has been absorbed, which leads to the electron from this state to the say fourth vibrational state of first excited state in electronic shell actually. So, these are electronic shells and these are vibrational, further division makes them into or divides them into vibrational labels, and further vibrational labels could be divided into rotational labels, which are not shown in here. And this distance is distance between electrons and nucleus, or we can say atoms, or we can say between atoms of a molecule or between atoms in a particular molecule. So, what you can see here is this is very clearly shown that the transition occurs from one electronic state to another. This transition could be between the vibrational labels of the two electronic states, that is ground state and first excited state, and rotational labels are not shown, which could be further division of vibrational labels actually. So, that is how. Likewise, if say this is transition from one label to another, but there could be like transition in within the electronic label between say vibrational labels. If I say here this is like changing to vibrational label, or in this particular excited state it could be like this could be returning into different vibrational labels. So, this is how a transition takes place between different energy labels actually. So, uh, I think it should be very clear that how how an electromagnetic radiation interacts with matter by absorption or emission, and electrons in atoms or molecules which resides in the ground state or lower level, by after absorption of the equivalent energy, that is the energy difference in the two labels, can get excited to a higher level. So, and the reverse will happen emission actually. So, a molecule that can be excited by absorption is known as chromophore actually. The excitation energy is usually converted to heat, say by collision of excited molecule with another molecule, or re-emitted as fluorescence. In both the cases, intensity of light transmitted by chromophore is less than the intensity of the incident light. Now, we will be discussing about the fluorescence spectroscopy in later lectures. Now, the major energy label, as we were discussing, depends on spatial distribution of electrons, and these are called electronic energy labels. So, what I have shown you, these were electronic energy labels, and I have shown you the ground state and first excited state. Then there are vibrational labels within electronic labels, or you can say in each electronic states, indicating various modes of vibration of this of the molecule. Now, these vibration this gives information about stretching and bending of various covalent bonds within the same electronic state. And then there could be further smaller subdivisions which were not shown in that diagram. These are called rotational labels, and they are not so much used in biology. In an energy label diagram, lowest electronic label is ground state and others are excited state, and as we have shown the first excited state in here. Now, absorption of energy can happen only if the amount absorbed corresponds exactly to the difference between the energy labels. So, light of a particular wavelength can only be absorbed only when there is a so, if you consider this, alright. So, amount could be absorbed only when like in terms of like say energy change for electronic transition, we can say what you should have is that change the difference in energy, that is between the two states, has to be equal to. So, the energy absorbed should be equal to the difference between the two energy states, that is the first ground state and then excited state, and also between the vibrational labels, to which vibrational label that particular electron is excited. Or in terms of like we can say that if terms of wavelength of electromagnetic radiation, then we can say that it should be that if it fulfills this particular one, that is again this component here, then the wavelength, the energy is will depend on the wavelength of electromagnetic radiation as well, and the wavelength at which the maximum absorption takes place is called lambda max. Lambda max means the wavelength at which a particular material or molecule absorbs the maximum, in terms like if I say in terms of absorption plot, if there is an absorption which is large absorption, there could be a minor absorption around here, but if there is a particular wavelength gives maximum absorption, then this wavelength will be called lambda max. So, a plot of the absorption versus wavelength is called an absorption spectrum, and this absorption spectrum comes from the absorption of particular wavelengths actually, which is the information that we obtain from absorption spectrum. Likewise, if it is a emission spectrum, then the spectrum, or it will be called emission spectrum, and there we will be looking for the emitted wavelengths or emitted radiations rather than absorbed radiations. So, these are two complementary techniques, spectroscopic techniques. Now, EM radiation or electromagnetic radiations, they are classified by the frequency of the radiation, or you can say in reverse it is a wavelength, but on the basis of frequency if we say, and they will be categorized into different types of electromagnetic radiations. These could be radio waves, microwaves, infrared radiation, visible light, ultraviolet radiation, X rays and gamma rays. and the total radiations including all these frequencies form the electromagnetic spectrum. Now, different radiations of the electromagnetic spectrum has been utilized in the biological science for variety of purposes. Now, if you can see on your screen, this is the particular diagram to depict various kinds of electromagnetic radiations, and as per their frequency, and that is increasing frequency or increasing wavelength. As the frequency increases, wavelength decreases and vice versa. So, if you can see in here, there are gamma rays at very high frequency, then X rays, and this frequency here is depicted in terms of hertz actually, and wavelength here is depicted in terms of meters. So, if you see gamma rays with very high frequency, then X rays, then next in electromagnetic spectrum is UV, that is ultraviolet rays. Then after ultraviolet rays, there is a visible spectrum, that is what we can see or the colors which we can perceive. Then comes the IR region or infrared region in the spectrum, microwave, then radio waves, and then long radio waves which are present in here. Now, if we consider like I said, there are different radiations of the electromagnetic spectrum have been utilized in the biological science for a variety of purpose. If I compare in here this one, if you can if little bit we can go into that, then UV and visible region, these are utilized, and little bit of IR region, these are utilized for as a standard what you call this particular region from UV till little bit of IR is for standard absorption spectroscopy. And here in absorption spectroscopy, mainly inner shell at the lower electrons and outer shell electrons are considered here for standard absorption spectroscopy. Then second is in IR region, you have like we have told you about vibrational and rotational thing. Now, in IR region there is a vibrational part is taken, and this is utilized for infrared. In vibrational region it is utilized for infrared and Raman spectroscopy. So, here in higher regions there will be nuclear magnetic resonance spectroscopy which will be utilized. So, these are like different, like another important thing is where we are utilizing in UV visible, where absorption spectroscopy is being used. This is in electronic label actually, where inner shell and outer shell electrons are taken. Infrared spectroscopy is performed as per vibrational shell here or vibrational label. These are types of transition I am talking about here. It is an electronic transition, these are vibrational transition, and as you go for say nuclear magnetic resonance, it is a nuclear transition we can say. So, these are different parts of the spectrum or electromagnetic, different types of electromagnetic radiations and the types of transitions we were talking about, and to how to what kind of techniques have been developed on that basis. So, in summary we can say that UV vis and even particular X rays are taken as standard absorption spectroscopy with electronic transition from X rays till UV vis and little bit of infrared. Then infrared is mostly vibrational transition, and it is utilized for Raman spectroscopy and infrared spectroscopy. And then finally, you have nuclear magnetic resonance, which is in say microwave region here, which is based on nuclear this resonance actually. So, these are different types of transitions which we were which have been utilized for obtaining information regarding the matter, that is interaction of the matter with the electromagnetic radiation. All right. So, as we have seen that spectroscopy is the study of interaction of energy with the matter and its analysis. and this has originated, this spectroscopy originated with the study of visible light dispersed by the prism according to its wavelength. So, like as we know that visible light is a combination of different wavelengths, and when they pass through the prism they are dispersed actually according to their wavelengths, as we have talked about this in microscopy also. So, in daily life also the colors of things can be taken as an example of spectroscopy, because objects absorb light of a particular frequency and emit a particular frequency and failing in the visible which are falling in the visible range and giving it a particular characteristic color actually, so that we can see it. So, this is like in daily life we are observing different colors because of like absorption and emission of a particular frequency wavelengths. Now, the principal concept of spectroscopy is resonance and the corresponding resonant frequency, that is it is resonating between the two different energy states. So, there is a major or principal concept is resonance and resonant frequencies. In quantum mechanical terms, resonance can be defined as the energy equivalence between two stationary states of a system, which is provided by the external energy source. Now, spectroscopy is a very wide field here, and where multiple subdivisions exist, and it is classified in a variety of ways, like nature of radiation, types of interaction and types of materials being studied. Traditionally, spectroscopy has been restricted to where particular absorption or emission takes place, that is a particular molecule or atoms absorb a particular wavelength or electromagnetic radiation of particular energy and gets excited, that information, or where emission takes place because of emission of a particular radiation, and those informations are taken, and that is a characteristic, or you can say traditional spectroscopy. And many techniques which might not have fallen in spectroscopy earlier have been regarded or considered in terms of spectroscopy, like for example, scattering actually, where or interaction in terms of not absorption or emission, but say in terms of scattering of light. That could be one form where that could also be called as a particular kind of spectroscopy, and as we will discuss as we go along here. So, it could be divided, like spectroscopy has become a very wide field, and there are a lot of multiple subdivisions, and it could be divided into various ways. For example, what are the nature of radiations are being used, what are the types of interactions which are happening, and what are the types of materials which are being studied. Let us take each one of them separately here. In terms of nature of radiative energy or radiations, which will include radiations belonging to different parts of the electromagnetic spectrum. Particles, it could be like electromagnetic spectrum, which as we have seen from right from X-rays till microwaves, and then ultraviolet and visible rays, infrared part of the spectrum. All these are utilized where interactions with matter takes place. Also, other than electromagnetic spectrum, there could be particles like electrons or neutrons, which could be taken, like there is electrons when they pass through matter, they could be scattered, or there could be other interactions. Likewise, neutrons can interact in various ways, and at times it could be also say waves, like say pressure waves or others used as energy source for spectroscopic studies. So, it is not a typical only electromagnetic spectrum or electromagnetic radiations which are interacting. It could be in the form of particles like electrons and neutrons as well, where scattering or other interactions might occur, will also be covered under spectroscopy. Then there are types of interactions actually, Now, there could be different types of interactions that will form the basis of spectroscopic technique actually. And as we were talking, the traditionally absorption and emission were the main constituent or main types of interactions which were considered or covered under spectroscopy, but there could be lot of other interactions like scattering, which could be elastic scattering and reflection, or we call it diffraction in many terms, like X-rays and electron scattering in crystallography, like X-ray crystallography, where electrons like X-rays are diffracted by the molecule which are arranged in a crystal in a particular regular array. So, that could be elastic scattering could be one form of interaction which is also comes under spectroscopy. Then there could be inelastic scattering, where it could be like it might involve exchange of energy between the radiation and the matter, and that will shift the wavelength of the scattered radiation, and it will include like Raman and Compton scattering. So, there could be both elastic scattering like in X-ray crystallography or X-rays and electron scattering in crystallography, or it could be inelastic scattering where change in or shifting of the wavelength of the scattered radiation could occur, and that includes Raman and Compton scattering. There could also be coherent or resonance spectroscopy. In coherent or resonance spectroscopy, like radiative energy couples the two quantum states of the material in coherent interaction, and that is sustained by the radiating field. One example is NMR spectroscopy is widely used method and is typical example of resonance method. There could be different types of interactions, right from absorption emission, which are traditionally considered, and then could be scattering, that is elastic scattering or inelastic scattering, and then there could be a resonance spectroscopy or interactions as well. Apart from types of radiation or types of interaction, also one other thing should be considered in here when we are talking about spectroscopy, that is types of materials which are involved in here. So, the spectroscopic techniques are designed to interact with specific kinds of materials, which could be like it could be atoms only in terms of atomic absorption and atomic emission spectroscopy. Like we will be talking about atomic absorption and emission spectroscopy, where absorption and emissions often referred to as atomic spectral lines, and these atomic spectral lines are due to the an electronic transition of an outer shell electron to an excited state, and atoms of different elements have distinct spectra, or we can say spectral lines. On the basis of these spectral lines, where it could be spectral lines in terms of absorption of particular lines, or it could be in terms of emission lines, emission spectra, where particular wavelengths have been emitted actually. Both ways these are characteristic of a particular atom, and these are very helpful in recognizing or identifying the particular types of elements or atoms in these sort of spectroscopy. So, one is interaction could be with like materials like atoms. Another could be interaction with the molecules. Molecular spectra can be obtained due to electron spin states, like where could be one is electron spin states could be there. Another could be molecular rotations actually, which could also utilized for deriving information. Then molecular vibrations which occur all the time, and electronic states. So, spin states could also be utilized, and electronic states which we have discussed earlier could be utilized. Then there are vibrations are which are also happening, and these are relative motions of the atomic nuclei and are studied by both infrared and Raman spectroscopy. Electronic excitations, as we have discussed, are studied using visible, ultraviolet and fluorescence spectroscopy. Whereas, for example, there could be, so we have discussed about atoms, molecules, there could be crystals as well actually. Like I said, these are regular arrangement or regular array of atoms, the regular arrangement of atoms in a particular array in crystals, and these regular lattice structure of crystals will scatter X-rays, electrons or neutrons, and these are utilized for crystallographic studies, where structure, three-dimensional structure of molecules could be obtained by generating the electron density maps. So, this is like a particular way, that is a scattering method could be utilized for crystals for deriving information, that is structural and chemical compositions. Then nuclei of matter could be interacting, like nuclei also have a distinct energy states and distinct nuclear spin states, and these can have their energy separated by magnetic field, and which forms the basis for NMR spectroscopy. So, in NMR spectroscopy, it is the nuclear resonance which is utilized, or nuclear magnetic resonance which is utilized for deriving the information. So, these are different ways, like, or you can say different types of spectroscopic techniques which are or the basis on which these spectroscopic techniques could be put in, and different ways like of interaction or types of radiation or different types of matters are discussed in here. So, all right, this was the basics, certain basic concepts in terms of electromagnetic radiation, in terms of types of interactions which are happening and how this information could be derived. What are the different spectroscopic techniques which have been developed, like on the basis of electronic transition, or on the basis of say vibrational transitions, or say nuclear transitions. The techniques like absorption techniques, or say infrared technique spectroscopy, or say nuclear magnetic resonance spectroscopy have been developed. Also other techniques which will be included in here, like X-ray diffraction or X-ray scattering, and mass spectrometry, which is not a typical spectroscopic technique, as we have explained. And traditionally, it is not a typical spectroscopy technique, but it is a very useful, very powerful technique, and is important. So, we have included it in here. So, the techniques which are a little bit into the techniques which we are going to discuss in this section here. One is ultraviolet visible absorption spectroscopy, which we are going to discuss here. In here, when electromagnetic radiation passes through a transparent material, a portion of radiation may be absorbed, and as a result of energy absorption, atoms or molecules will pass from a state of low energy, that is ground state, to state of higher energy, that is excited state. And ultraviolet visible spectroscopy is based on the transitions of electrons from one atomic orbital or molecular orbital to another, and due to the absorption of the electromagnetic radiations. So, we are going to discuss in detail about this technique in coming lecture. Then another based on vibrational transition is infrared spectroscopy, which deals with the infrared region of the electromagnetic spectrum, having higher wavelength and lower frequency than visible light. It consists of three regions, that is near 0.8 to 2.5 micrometer, mid 2.5 to 25 micrometer, and far infrared, that is 25 to 1000 micrometer. And based on the fact that molecules absorb specific frequencies in this range that are characteristic of their structure, and the absorbed frequency resonates within the frequency of the vibration of the group. So, the IR active molecules require a change in the dipole moment The IR spectrum is recorded by passing a beam of infrared light through the sample, and when the frequency of the infrared beam is equal to the vibrational frequency of the bond, IR absorption occurs. Then next technique we are going to discuss about is fluorescence spectroscopy. Fluorescence spectroscopy is a type of spectroscopy where fluorescence from a sample is analyzed. The phenomenon involves the absorption of light, usually in ultraviolet region, that will excite the electrons in molecules of the certain compounds and will cause them to emit light at a longer wavelength when they come back to the ground state, usually in visible region. So, that we are going to discuss in coming lectures. Then also we are going to discuss, we have included certain other techniques like which are typical spectroscopy techniques, of course, like circular dichroism, which are very useful in various biophysical applications. Circular dichroism, here the electromagnetic radiation consists of an electric and magnetic field that oscillates perpendicular to each other and to the direction of propagation. So, while linearly polarized light occurs when the electric field vector oscillates only in one plane, and circularly polarized light occurs when the direction of the electric field vector rotates about its propagation direction, while the vector retains constant magnitude. At a single point in space, the circularly polarized vector will trace out a circle over one period of the wave frequency, hence the name has been given. So, the circular dichroism refers to the differential absorption of the left and right circularly polarized light. The phenomenon of circular dichroism is exhibited by chiral molecules, and hence biological molecules are expected to exhibit the same due to their dextro-rotatory or levo-rotatory components. Moreover, a distinct secondary structure imparts a characteristic CD spectrum, and like alpha helices and double helices in DNA have characteristic CD signatures. The far UV CD spectrum of proteins can reveal important characteristics of their secondary structure, and CD spectrum can be readily used to estimate the fraction of molecule that is in the alpha helix conformation. So, the beta sheet conformation and also some other like say random coil conformation could be obtained. These fractional assignments place important constraints on the possible secondary conformation that the protein can be in. Then we are going to discuss about nuclear magnetic resonance spectroscopy, which exploit the magnetic properties of certain atomic nuclei to determine physical and chemical properties of atoms or molecules in which they are present. Method is based on the nuclear magnetic resonance and provides a detailed information about the chemical environment, reaction state and structure of the molecules. Basically, when a sample contains NMR active nuclei, for example, 1H or 13C or 19F or 31P, these have fractional spin of half, and when they are placed in magnetic field, they exist in two spin states, plus half which is aligned with the magnetic field and minus half which opposes the magnetic field, and a difference in energy between the two spin states will depend on the external magnetic field strength. When these are irradiated, sample is irradiated with the radio frequency energy which corresponds to the exact spin state separation of the specific set of nuclei, that will cause excitation of nuclei from plus half state spin state to higher state, and the resonant frequency, absorption and the energy absorption of the energy and the intensity of the signal are proportional to the magnetic field. There could be lots of different kinds of NMR techniques, which could be one dimensional techniques for smaller compounds to 3 or 4 dimensional techniques for large molecules like proteins or nucleic acids. Another technique we are going to talk about is X-ray spectroscopy, and we will be more talking about X-ray scattering or X-ray diffraction methods. Now, X-ray spectroscopy refers to the spectroscopic technique for characterization of different material using X-ray excitation. Now, when an electron from inner shell of atom is lost due to some sort of excitation, then vacancy is filled with an electron from the outer shell, and the difference in energy is emitted as X-ray of appropriate wavelength characteristic for the element. So, the analysis of spectrum of X-rays provide qualitative information about chemical composition, and comparison of spectrum with spectra of standards of known composition provides quantitative results. Now, X-rays can be absorbed by matter also and gives rise to X-ray absorption spectra. Then there could be X-ray fluorescence analysis also using X-ray fluorescence. Apart from this, there are X-ray scattering or diffraction techniques which reveal information about the crystal structure or structure of the molecule, and these techniques are based on scattering, or we can say diffraction of an X-ray beam by crystals at different angles and intensities, and from these angles and intensities of the diffracted beam, a three-dimensional distribution of density of electrons within crystals is obtained, and therefore, the mean positions of atoms in the crystal and the structural information can be derived. So, we will be dealing about this in later chapters. Then also we are going to discuss about atomic absorption and flame emission spectroscopy. Atomic absorption and flame emission spectroscopy methods are very powerful tools for both qualitative and quantitative analysis. In atomic absorption technique, absorption of radiant energy by free atoms in gaseous state occurs, and that is the basis for obtaining the information, whereas in emission or flame emission technique, there is intensity of light from a flame or plasma at particular wavelength is emitted, and this gives information. Wavelength of atomic spectral lines helps in identification of elements, while intensity is proportional to the number of atoms present. Finally, we are going to discuss about mass spectroscopy or mass spectrometry. To say, mass spectrometry is an analytical tool used for measuring the mass to charge ratio of the sample that are charged. The instrument that do for same are called which does this is called mass spectrometers, and which has ionizer, analyzers and detectors. Here the sample has to be introduced into the ionization source of the instrument. Once inside the ionization source, the sample molecules are ionized, and because ions are easier to manipulate than neutral molecules, these ions are extracted into the analyzer region of the mass spectrometer, where they are separated according to their mass to charge ratio, and the separated ions are detected and the signals sent to the data system, where the these ratio m to z ratio are stored together with their relative abundance for presentation in the format of a mass to charge ratio spectrum. The analyzer and detector of the mass spectrometer and often the ionization source too are maintained under high vacuum to give the ions a reasonable chance of travelling from one end of the instrument to the other without any hindrances from air molecules. Entire operation of the mass spectrometer and from the sample introduction process also is under complete data system control on modern mass spectrometers, and this technique is utilized for various things like determining masses of particles, for determining the elemental composition, for elucidating the chemical structure, etc., and this also we are going to discuss in coming chapters or coming lectures. In this lecture, to summarize, we have discussed about basic concepts in terms of electromagnetic radiation and the spectroscopy, which is the interaction of the matter with the electromagnetic radiation. We have discussed about different types of spectroscopy and the basis for those spectroscopy techniques like electronic transition or vibrational or say nuclear resonance, and a lot of different spectroscopic techniques have been developed. The area of the spectroscopy also has been widened to include more spectroscopic techniques like on the basis of scattering and other phenomena. So, we will be starting with the specific techniques in the next lecture, and in the next lecture we are going to discuss about the first technique, that is UV visible spectroscopy. Thank you.